Okay, let's start. Good morning, afternoon, and evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa. 안녕하세요. And hello all. Welcome and thank you, SynBYSS people, for joining us, the 173rd seminar, and I call it a National Institute of Health Day. On September 4th, 1998, I mean exactly 27 years ago from today, Google was founded by two PhD students, I mean young people at Stanford University at the time. As you may know, Google Search and YouTube are the most visited sites revolutionizing our daily life. For example, we used to gather information from libraries or by asking people and listen to music using CD or tape players, while we now just Google it or ask AI and listen to or watch YouTube videos, essentially as long as you have internet access or phone, tablet, or computer. The global distribution of this seminar series talk videos is also possible thanks to the YouTube platform. We are working on synthetic biology or engineering biology problems, and I hope young researchers or scientists and engineers, I mean you, will become visionary people like the Google founders, revolutionizing the biotechnology and synthetic biology field and the society as well through your imagination and innovation. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Dr. Tuba Fehr. She is a program director at National Institute of Biomedical Imaging and Bioengineering, in short, NIBIB, National Institute of Health or NIH. She has been shaping the future of bioengineering and helping researchers develop innovative and significant technologies for potential medical applications. I believe I met her a few years ago for the first time, and she kindly accepted my invitation to speak for this webinar audience. I also found she is one of the kindest program directors, guiding researchers to write great proposals. Tuba, thanks so much for your support to the research community, and the virtual podium is all yours now, and thank you again.
Thank you so much, Taesuk, for your very kind words. The honor is mine to be a part of this effort in being a part of this community and helping shape the future of synthetic biology, as you mentioned. And I just want to say in your string of hellos, I think you missed out on the Turkish hello, which I would like to say merhaba to everybody, because I know there are Turkish synthetic biologists out there, and I'm originally from Turkey. Just wanted to put a plug in there. But it's my great pleasure being here today, and I'm gonna do my best in summarizing NIH to everyone in about 10 minutes. So it'll be fairly quick. But I hope you reach out to me directly after this presentation so we can have more extended conversations about your projects and how to apply for NIH funding. This is just an overview. There's a lot of nuances in how to get NIH funding successfully, so I'm happy to take the time and guide you through this maze of NIH funding application process. So I'm going to share my screen now for my presentation. Okay, do you see it in presenter's mode? Okay, perfect. So yes, as Taesuk mentioned, I'm a program director at NIBIB at the NIH in the Division of Discovery, Science, and Technology, which is bioengineering. And sorry. Okay, just a brief overview of synthetic biology at NIBIB. We are currently 3 program directors that cover the SynBio portfolio, but mainly it's me and my colleague Nicole that divide up the portfolio in 2 ways. Mine is the cellular and multicellular technologies. These are synthetic biology tools and approaches that focus at the level of cellular engineering or organoid or tissue level engineering, multicellular level. If you're working on tools and approaches at the subcellular level, like new CRISPR tools or any genome engineering, that would fall under my colleague Nicole's portfolio. So it's always advised that you reach out to one of us so we can, better guide you as to whose portfolio your ideas may fall into. And I'm putting in here, I'm not going to go into details, I'm putting in the website here for our division so you can go and look at the different portfolios and see the areas of interest for NIBIB when it comes to synthetic biology and other portfolios. And I believe my colleagues, Jermont Chan and Dave Rampula, have presented before, so they are also part of my division, and we're all happy to chat with you about your NIH applications. So I'm going to give you a brief overview of the NIH for those of you who may not be familiar with it. NIH, first of all, I would like to give you the overview of NIH, and then the question is, NIH is a big entity. So which institute, it's composed of many institutes, which one to apply to with your grant application, what funding mechanism to apply to, funding mechanisms specifically geared towards early career stage investigators, and also some tips on how to reach out to program officers to discuss your ideas and strategize for best way to put together an application for the NIH. So as I said, NIH is the largest public funder of biomedical research in the world with a budget of $49 billion to enhance life and reduce illness and disability through medical research. A substantial portion of this budget, about $35 billion, is issued via grants to thousands of entities, including universities, hospitals, and small businesses and organizations to conduct biomedical research. NIH has 27 institutes and centers, so it's a big entity. And among the 27, 24 of these institutes give out research grants. The 3 that are shown at the bottom do their own, they have other missions and other activities, but they do not give out research grants. And as you see here, I'm from NIBIB, the National Institute of Biomedical Imaging and Bioengineering. We are one among many of these NIH institutes that fund biomedical research, including synthetic biology. It's not only NIBIB that funds synthetic biology. There's a bunch of us. We actually have a synthetic biology interest group at the NIH. So there's a bunch of program officers very passionate about SynBio, and we get together and talk about how to help the community and stimulate research in this area. Now, you may be looking at this chart and you may be a little overwhelmed. So, you have this brilliant idea that you want to pursue, but research money doesn't grow on trees. So you need to look for a funder to fund your project. And you're looking at this chart and saying, where do I even go? So I'm hoping to demystify that a little bit and give you some tools so you can, start the process if you're not familiar with the NIH at all. There is a wonderful tool called the NIH Matchmaker that you can use to pinpoint which institute may be a good scientific home for your research project. The Matchmaker tool allows you to put in your specific aims, abstract of one of your papers, or even a few keywords, and you can use the search box to find similar projects that have been projects similar to yours that have been funded by the NIH previously, which institutes have funded those type of projects, which program officials oversee portfolios similar to the projects that are similar to yours, and also some study sections that review projects that are similar in your field. So this is a great tool. The link is there. Just go and play around with it. I use it for my own work all the time. This would give you an initial entry point if you do not know where to start, which institute, which program officer to go to. If you have no idea, plug in some keywords, your specific aims. It does not save any of the input there, so do not be afraid to put in your specific aims page. Obviously, do not put confidential information in there, but you can paste in whatever you want, and then it will direct you to a list of potential institutes and program officers to reach out to. Now, you've identified which institute may be a good fit for your idea, but what mechanism? There are more than 200 different grant mechanisms and activity codes at the NIH. It's pretty overwhelming. You don't need to know all of them. That's our job, to guide you to the correct one. Here's a link and a list of potential funding mechanisms. So it depends on the nature of your work, your eligibility, and other considerations. So it's always best to check with a program officer ahead of time to see what mechanism may be a good fit for your project at hand. In general terms, there are 4 types of mechanisms that we use at the NIH. The first 2 are geared more towards young investigators. So these are the fellowships at the pre-doctoral level or the postdoctoral level. There's the Career Development Awards for advanced postdocs and early faculty members. There's institutional training grants where established PIs can use these training grants to fund trainees under training slots, and also the research project grants that people are familiar with. These are the Independent Research Awards. When you establish your own lab, you're an assistant professor or an established professor, you have a new idea, you apply to a research project grant to fund your ideas for the continuity of your research program. So you can find more info under the link here when it comes to research training programs. Again, there's many different flavors, so it's always best to check with the program officer. Now, in terms of individual fellowships, there are, again, different flavors based on your stage and career trajectory. There is if you're a PhD student, there's the F31 fellowship. If you're a postdoc, there's the F32 fellowship. And if you're a PhD level MD PhD student, you can apply for the F30 mechanism. The only thing to keep in mind about the fellowships is that you cannot apply for an individual fellowship if you're on a visa studying towards your degree. Unfortunately, you either need to be a citizen, a non-citizen national, or a permanent resident for eligibility purposes to apply for individual fellowships. So unfortunately, those trainees who are studying in the US on a visa are not eligible for this. Our contact at NIBIB for fellowships is Dave Gutekunst. Please reach out to him and he'd be more than happy to talk over eligibility criteria and how to put together a competitive application. For later stage, more advanced postdoc or level trainees, we have career development, K99 R00 awards. This is a biphasic award, meaning there are 2 phases to the project where the initial 2 years, we call the K99 phase, is done during a mentored postdoc fellowship. And then after that, you continue your line of research as an independent faculty for 3 years as an R00 phase. If you are ineligible if you hold any major NIH grant or career award, this is purely geared towards early career investigators. This one, contrary to fellowships, do not have a US citizenship requirement. So any postdoc or early career investigator can apply. Our contact for this mechanism is Tina Gatlin at NIBIB. Again, she'd be more than happy to talk to you about the K99 awards. A couple of other mechanisms that I'll mention that are geared more towards early stage investigator is the NIH Director's New Innovator Award and Early Independence Award. These are under the High Risk, High Reward program run by the Common Fund at the NIH. And these are for exceptionally innovative research from scientists at all career stages. Again, the New Innovator and Early Independence Award are geared towards early stage career folks, but we have different varieties for even established investigators. These need to be more groundbreaking research that may not fit traditionally into an R01 mechanism, for example, that are typical of NIH programs. One program area that I thought would be helpful to highlight to this group is the loan repayment program. This is a program that maybe not many of you are familiar with. And this is a great program. And the intent is to recruit and retain highly qualified professionals into biomedical research careers. And if you have an MD or PhD or an equivalent and you're a US citizen or a permanent resident, you can apply for the LRP program to get funding to do research. I am not familiar with the nuances for this. Again, our contact is Tina Gatlin for this program. But essentially, you do not need to have NIH grant support to apply for an LRP grant. It essentially pays for your educational debt. These are 1 to 2 year contracts, $50,000 annually, that you can use towards paying your educational debt down. In exchange, you are expected to dedicate at least 20 hours per week on research that falls within NIH mission. Again, I would encourage you to reach out to my colleague Tina to inquire about this program, which I think is a great program. And finally, I'd like to put a plug in for our NIBIB specific early career stage Trailblazer Awards. This is an R21 mechanism. I'm not going to go into the specifics of what R21 is, but just to highlight that these are exploratory / developmental grants, meaning new bold ideas, never been tested before, high risk, high reward type of approaches. The key about this award is that you are only eligible for a Trailblazer if you're a new or an early stage investigator. Early stage investigator for the purposes of NIH is anyone who is within 10 years of their terminal degree, their PhD. New investigator is someone who has never had a major NIH award before. So you could be a professor, a late career professor, but if you've never had NIH grants before in your career, then you are a new investigator. You're still eligible to apply for a Trailblazer Award. There is no preliminary data requirement expectation. So this is a great way to test some bold ideas and take some conceptual risks and try something new. This award is meant to allow you to generate preliminary data for and allow you to have that solid foundation to apply for other mechanisms such as R01 later down the line. So if you're a new professor, assistant professor, you can apply for a Trailblazer Award. It's protected time for 3 years, $400,000, and you can do your research to generate that preliminary data to launch your research program. So one last thing before I go, it's a lot of information, and if you're overwhelmed already, don't worry, just reach out to one of us and we're happy to talk to you. I spent hours talking to investigators about the NIH, and I'd love to do that with you also. So always reach out to, if there's one take-home message you have from this presentation, is that always reach out to an NIH program officer ahead of time. Do not wait till the last minute. I've had people contact me 2 days before application due dates, and I'm like, I really cannot help you as much as I could have if you contacted me a month ago. But reach out to program officers using, identify them using the NIH Matchmaker. And when you reach out, be mindful to include some information so we have an idea of who we are talking to and how we can best help you. Include your academic background. Are you an engineer? What kind of scientist are you? What are your current research areas? What career stage are you? And it's always helpful to include a specific Inst page and a CV for us to have a better sense of who you are. Your shared goal really in reaching out to us is to figure out, again, what grant mechanism, there's so many of them, which NIH Institute, there's so many of us, whether you're eligible and when to apply. Again, it's encouraged, it's advised that you reach out a month before the due date, but reach out regardless, even if it's closer to the due date, reach out, we'll do our best to guide you. Some best practices and do's and don'ts for when reaching out to program officers. Again, use the Matchmaker tool. 1 month before the application due date, reach out. it's always best to have your specification page ready before you reach out, because if you reach out and say, I have this great idea, I would still ask for a specification page before I schedule a call with you, because it's best. I like giving more directed guidance. So I need to see the scope of your project to make a determination if it's a good fit for an NIBIB mission or whether it should go to a different institute. So it's always helpful to see the specifics of your project. And also, please reach out to a single PO at an institute. Do not blast emails to 5, 6 different program officers within the same institute. We all know each other. And then if we don't know that you reached out to us individually, then we're all reading the same thing and then finding out that, all of us have actually independently tried to reach out to you, which is just not very efficient. So reach out to one person, and then we'll be happy to direct you and triage from there. I think that's all I have. This is my contact information. If you have any questions, I'm happy to follow up after this talk or anytime. Use my email. And thank you so much again for the opportunity, Taesuk, and it's been my pleasure.
Thank you so much. This is a wonderful information. Also, I want to thank all NIH. Actually, NIH is the largest number of program officer already giving a talk. Also, will give a talk for this seminar series. And total number up until now, I believe 7, including Alpha H, actually 8. So largest number along with the NSF. Also, NIH also kindly supported the conference last year. So I hope, the NIH continue to support young people. And a lot of speakers in this series also got the NIH grant. So I'm very grateful for your support for the community. Thank you so much.
Thank you.
All right. So now the main speaker of today with a longer introduction, Dr. Shohei Kitano, on his PhD from the Tokyo Institute of Technology, where he was studying the non-coding regions of the human genome, specifically focusing on the 5' untranslated regions of mRNA. He is currently working at the National University of Singapore under the guidance of Professor Matthew Chang. He's the world leader in medical study. He was involved in the SC 2.0 project. I believe many of you know about that project, which aimed to synthesize a whole genome of Saccharomyces cerevisiae. This is actually international collaboration project with gigantic impact to the community. He is interested in the design principles of genomes. This interest began during his PhD, where he examined the functions and evolutionary origins of non-coding regions. During his PhD, he realized the limitations of reductionism in fully grasping genomic intricacy. This led him to explore synthetic genomics, focusing on redesigning the yeast genome. Currently, he is interested in applying a synthetic genomics approach to understand the phenotypes caused by multiple gene variants. Shohei, thanks so much for your time today, especially joining us from uninconvenient time zone. I believe the past
Yeah,
now
11:30 now.
11:30 p.m. I believe. And
Yeah.
please take it away. Thank you so much again.
Okay, yeah, thank you very much for the kind introduction. And yeah, I'm so surprised why I, just now introduced myself as totally same as what you used. Okay, anyway, so okay, let me share my slides. Share. Okay. Can you see this, right?
Yes, go ahead. Thank you.
Actually, now how many people on the live? Okay, so thank you very much for introduction. I'm Shohei Kitano. Now I'm working under the Matthew Chan, and then now I'm currently working in National Center for Engineering Biology. This is a new center. I think they established last year. I think now it's the second year. Okay, so and then this is our lab. Chan lab is doing the many. Actually, we are doing synthetic biology, and then we have three pillars. And the one is the sustainable bioproduction, so doing the metabolic engineering for the sustainable production. And the second one is therapeutics way, so they engineer the bacteria for the therapeutic purposes. I think a few months ago, the one of my colleague, Hao Shen, also came here to talk about the current publication data. And then third one is the synthetic genomics, which I'm going to talk about now. And then as I introduced, we were working on SC2 project. And then yes, in the project, we synthesized the entire chromosome 15 of the Saccharomyces cerevisiae. Yes, but we didn't just synthesize the same sequence as Y-type chromosomes. We actually added a lot of the recoded regions. The one example is that we added what can I say? Yeah. We added PCR tag is the unique watermark sequences. So we added 1,408 recorded sequences. So like this. So the PCR tags allow us to differentiate the Y-type sequence and synthetic region. So one of the purpose is that we can confirm the synthetic DNA is really replaced the Y-type chromosomes. And second one is remove the TAG codon to TAA codon, so that in future, once we synthesize all chromosome, TAG codon is not used as stop codon, so we can add 21st amino acid for the codon tables. And third one is we insert the LOXP sim site at the end of all non-essential genes. So this allowed, upon the expression of recombinase, the LOXP recombination in synthetic chromosome can make many variety of the chromosomes. And the next is we remove the tRNA and the repeat sequences to stabilize these synthetic chromosomes. And finally, we deleted all introns from the messenger RNA. So all genes in the synthetic chromosome is intronless. Yes. And then we synthesize 1.0, almost 5 megabase DNA in total. And then today I'm going to talk about the how to synthesize and how to assemble and how to find the deficit region of synthetic DNA and how to apply the synthetic chromosome to elucidate the biological mechanism. Okay, then I'm going to talk about how to synthesize the chromosomes. So firstly, we chemically synthesize fragments of 5 to 6 kb, and we in vitro digest it and ligate it, and to create 20 to 30 kb DNA. And this DNA was introduced into the yeast cell and integrated and replace the Y-type sequence to the synthetic regions. And then we put the markers at the end of the ligated DNA. So every time we can swap and replace the marker gene. So we just keep doing this and then we can elongate the synthetic regions. But actually this experiment very easy. But actually getting the real one is very hard. So every time actually we do the 2 marker genes, but sometimes it's partially integrated. And then we have to check every time by the PCR to make sure that the clone has the fully synthesized regions. And after doing this, but the issue is doing this from the left to right takes a very long time because we have to do this 46 times. 46? Yeah, around 46 times. So it takes a very long time doing the 1 by 1 because one cycle takes like 3 weeks. So we didn't do it. and then we make the partially synthesized chromosomes. And then we integrate these into one chromosomes by consolidation. But there was an issue, this methodology, because there was conventional method is just made them. And then they just the hoping some clone as the fully synthesized one after aspiration. But this is getting more and more difficult as the synthesized DNA become bigger. Like in our case, we have to make a 1 megabase DNA. So during spiration, and the many recombination site happen between the chromosome 15 and the 15, and then we always get the white type chromosomes. So we needed to develop the one rational recombination method. Then we develop the creamier that allow us the site-specific reconfigure chromosome, and it's quick screening. The trick is after mating, we just cut the CRISPR here. We target this region with CRISPR and then get the homozygote diploid and then sporulate to get the fully synthesized haploid yeast cell. And this, I'm going to explain about the detail of this methodology. We named this method creamier, standing for the CRISPR-Cas9 mediated mitotic recombination with endo-rib duplications. The first step is that we prepare two cells, each partially synthesized, having the partially synthesized chromosomes, having a guide RNA expression vector or Cas9 expression vector, and then cross them and then make the diploid cell, having both guide RNA and the Cas9. So the CRISPR become active. And they start we design the guide RNA cutting the boundary between these two and here, and then activate the recombination between the two chromosomes. But resultant one is not the perfect. So after mitosis, we sometimes get those kind of one. The trick is you can see the red dot one. This is URA3 marker genes. We can do the negative selection. And then another point is we put the galactose 1 promoter stream of the centromere. So upon the induction of galactose, the galactose promoter is inactivate centromere function. So this chromosome is sometimes segregated randomly. So some cells lose this chromosome, but this chromosome. And then by combining galactose induction and 5FOS selection, which is select the URA3 negative chromosome, and we can get this 2n-1 diploid with a single copy of SYN15. And then this is always the grow slowly. So, and it causes endo-reduplication, and we can get the two copy of SYN15 in diploid cell, and we can get the SYN15 haploid cell by spiration. So this is the mechanism of the creamier we invented. And then first we tested using this partially synthesized one in the left arm. We call this AC1A and AC1B. And then we put the galactose promoter here. And this red one is URA3 markers. Yeah, as I explained, just cross it, and then we can get this one. And then the efficiency is quite high, surprisingly. And then we we conducted the PCL. So this the prime we recoded the gene, synthetic gene. This allowed the designing the primer specifically bind to the synthetic genes. And then we can also design the primer specifically binds the wild type. That's right. So that we can differentiate which strain has the fully synthesized or which strain still have the wild type sequences. And this data is showing that we first tested the PCX, this region, A2 and M4 region. And you can see that all clone tested has the synthetic regions. So this means the all tested 10 is showing that actually happened the recombination. But recombination point is always not accurate, even using CRISPR. Even we design here, sometimes recombination happen upstream or downstream. So we have to the screen the clone that actually happen the cutting point. So we conducted a PCR, target PCR to the region, the eight regions, no, 10 regions. And then this PCR, this is a result of the PCR, attack PCR. You can see most was synthetic, but some point like this clone 4 has the wild type sequences. Yeah, and the 7A or yeah. But the point is just testing Tang, and then we can get the few correct colonies very easily. This is much better than the conventional method. I think it's almost impossible to take the fully synthetic one, just taking 10 or 100. I think have to test really 1,000 clones. Okay, and then we use this methodology to integrate the assemble the fully synthesized chromosomes. Yeah, and then this is the result of the PCL. You can see that this is the syn tag and this is wild type tag, and then you can see all bands in the upper gel. Yeah, so we tested all PCR tag, and then we confirmed that everything was replaced to the synthetic region. And then this is a post-PCR electrophoresis. So this methodology can separate the chromosome level DNA using agarose gel. And you can see that the syn 50. This band is representing the chromosome 15 and 7. In wild type, the two chromosomes are overlapping, but synthetic 15 is smaller than the chromosome 15 because of the deletion of the tRNA and the repeat sequence and intron. You can see the size reduction of the syn 15. Of course, this doesn't say that the fully synthesized under the sequence error. So we perform and actually during the process of assembly, sometimes it causes a duplication of some regions. So we perform the biosequences and de novo assembly. And then we align the de novo assemble sequence to the sequence we design. And you can see this one dot line, meaning that this completely the match to the original one. There is no duplication in our synthetic chromosomes. Yes, then we successfully synthesize the sequence we designed. However, actually, the first version of syn50 shows several defects. First one is actually very slow growth. Like I think the 20 or 30 minutes longer than the wild type original one. The other issue, this is a very big issue for the genetics, that it couldn't sporate. I think sporation rate was very low. Almost cannot see the sporated cell in the normal conditions. Yes, and then so we hypothesized we can apply the creamier, which now we use to the assembly the chromosome. We can use this methodology to identify the causal recorded sequences that these defects. And then applying this creamier and the DBTS cycle, and we can identify the causal region systematically. This is a schematic to explaining this. Okay, now when we assemble the chromosome, we target the boundary here. But by changing the guide RNA sequence, and we can target like here or here or here. By changing the target site, we can get the combination of reconfigured chromosome like this. In this figure, the green bar is indicating wild type sequences. You can see it's not everything blue. And we can change the region of the wild type by changing guide RNA sequences. So we can get the combination of the reconfigured chromosomes. And then we do the phenotyping testing. And then we can learn which one is actually still have deficit and then which chromosome doesn't have it. And then we can narrow down the responsible region and then just grouping this cycle, and then we can identify the gene level and then record the region level. Like this. This is the first example. We first test the left arm of the synthetic chromosomes. You can see by from these three actually grow faster like the wild type chromosomes. But this last one is obviously showing the deficit like this. This is the last one. So this can be, from this data, we conclude that this region is actually causal regions. This is the growth defect. And the next round, we change the guide RNA to cut this region. And then we identify this region in the responsible region. I cannot see that. Okay. Yeah. And then okay, and after the identification of the gene level, next we check the TAPICIA level. So this is one gene, IRA2, and then there is the red neighbor gene REX4. So this is the candidate genes causing the issues. And then we have to identify which recoded sequence is actually causing the growth defects. And this green, everything green means everything white up sequence. This is it's much faster than this synthetic 3.1. Yes, it's around 20 minutes faster in doubling time. And this is also fine. And then commonly the synthetic region common region is causing the bag is this region, is the lesion 8. You can see this is slow and this is also slow and this is also slow. So we hypothesize this region is the causing region. And next what I do is RNA-seq. We perform RNA-seq and then we also find these genes, the differential expression gene pattern is very close to the strand lacking these genes like this. So you can see the trend. So this is indicating our synthetic 15 strand is a similar gene expression profile to the strand lacking these genes. So we hypothesize that this region is actually repressing the expression of these genes. And then this is the result of RNA-seq map onto this IRA2 genes. You can see this is the BY4741. You can see just the green birds. And this is 3.1, the first version of synthetic 15. And then the bar is going upward is the whole transcription. And the bar going down is representing the translation on the reverse side. So this RNA-seq data is showing that somehow the species tag is created by directional promoters, and then probably this anti-transcript repressing the expression of IRA2 and then causing the issue of the phenotypes. And IRA2 is known as a GTPase activated protein, and then this negatively regulates the LAS to reduce intracyclic AMP level. So actually, the repression, this IRA2 is hyperactivate the PK pathway. So this very makes sense that there's phenotypes as well. So to validate this hypothesis, we designed CRISPR-I. So because there is anti-transcript, so our hypothesis, if we use CRISPR-I and repress this transcription, this anti-transcript, the cell growth also recovered. And this is the result of the CRISPR-I. This is the antisense transcript. And then this is the mark, and this is the guide RNA. And when we treat with this CRISPR-I, the antisense transcript reduced. And at the same time, IRA2 expression also increased. So this data is strongly showing that this antisense is the repressing expression of the IRA2. And at the same time, the growth also become faster by the IRA2. So, IRA2, the sgRNA. So we conclude that somehow the recoded sequence is created promoter and repress IRA2 expression. So when we do synthetic chromosome stuff, we can often encounter those kind of unexpected stuff actually. Yeah. Okay, and then this is the first bug we identified. And then next round, and then we use the premium again. The previous one is we targeted the left arm, and next time we target the right arm. So design the two guide RNA first, and then we make the two types of the new synthetic chromosome. And then at this time didn't change a lot, but we identified this region is also the causing the growth defect. Yeah, especially this height. And then we did another round of DBTL by changing. No, not this time. Okay, so we identified this one. So next is we upgraded the Crimean. This method, we can change the terminal sequence. But we wanted to change, for example, we want to just change this region only. So we use the two-guide RNA at the two site, and then we can just swap only these internal regions. And then we made this one, and we also showed that by changing this region, that the growth become better. Yes, and then we crossed refer the RNA-seq data, and then we found that two genes are actually differentially expressed in this region. And then next, and then OSW1, and then we changed both the genes to the wild type sequences, and then we observed the increment of the final ODs. And another phenotypes also has been improved by this. Actually, I see 2.0, we do the many spotting assay with under many stress, and then we actually found this synthetic chromosome has an issue using the glycol as a carbon source, indicating mitochondrial issue, by the way. And then, but after changing this bag, these two genes into the wild type, and we could see that the improvement of the growth on the YP glycol. You can see the difference is very obvious. Yeah, and this is how we identify the two bags. Yes, so yeah, I think it's not mentioned often about the synthetic chromosome project, but yeah, those kind of thing is happen very often because we changed more than 10,000 bases, so many unexpected reactions happen. So the dealing with this issue also the key in the synthetic genome project. The design sequence is not always work as we expected. So we developed the methodology to easy and systematically find a bag. This is one of the contribution to the study. Okay, and then we found another bag also using different approaches. I want to explain the detail of the RibeSec, but the technology is to monitor the translation status in vivo, and then we find a bit of the unique change in this genes. This gene is actually have open reading frames in the 5' UTL, and it is known that this UTL, this open reading frame, is also translated. But this insertion of LOXP is significantly increase the translation of this upstream open reading frame, and then drastically reduce the translation from the CPA genes. And so we hypothesize the LOXP, I think the LOXP sim is a palindromic structure. So I think inserting this sequence into the 5' UTL create a strong secondary structure. And then this, I think, slow down the ribosome. That's why I think the ribosome scanning is the speed reduce and this activate the translation from this upstream operating frame. As a result, after translation, ribosome, of course, release a messenger RNA. So this cannot read to the start codon of the CPA1. That's why I think insertion LOXP repress the translation from the CPA1. And then we also changed this to the white that we removed this LOXP sim site. And then we also confirmed the phenotype is drastically improved. So by doing this, the debugging process, that we improved the synthetic chromosome phenotype drastically. So this is a spotting assay and the YPD or the complete media, synthetic complete media, YP glycol or other under many stress conditions. You can see that we just please compare the left and the right, and then it drastically improve compared to the middle one. So many phenotypes is much closer to the wild type compared to the original one. So we show that this our methodology, and then driven the chromosome scale DBTL can improve they can easily identify the causal bag and systematically. Yes. Okay, so this is how we the synthesize and assemble all the debugging and the synthetic chromosome. From next, I want to introduce how we utilize the synthetic chromosome to elucidate the fundamental and the biological questions. And then an idea that we can use the synthetic yeast as a learning platform of translation mechanism. And then we conducted ribosome profiling. The first I want to explain about the ribosome profiling. Maybe many people know it. The ribosome profiling technology using the Illumina sequencer to identify which codon is translated in vivo. The mechanism like this is we add the cyclohexamide to stop translation in vivo, and we treat the RNA, these RNAs, but the RNA region protected by ribosome, meaning translated region, is not digested by RNAs. So this fragment is protected. And then we collect this protected sequence and a deep sequence and map the genome. And we can identify which region is actually translated and which region was A site at the time. Because this length of the protected RNA is totally depending on the ribosome size. So we can tell easily which size actually, which region is the A site, meaning the translate site, very easily. So we can identify the translate activity at the codon resolutions. And then by combining this, and then we thought that we can answer the question, is the codon usage a determining factor of translation rate? I think in synthetic biology, many people are always trying to codon optimize because the codon usage is a very important factor for the translation rate, right? And sometimes if you do not optimize codon, maybe some people believe that it cannot express properly in especially exogenous genes. And then actually it's not still controversial question. Then some people say codon usage is a determining factor of translation rate. And some people say actually the codon usage is not a determining factor of translation rate. But in many studies, those kind of things is relying on not many. Some people, those kind of things, determining factors saying just changing the one genes of the codon usages. Another one is that doesn't change it, but by comparing the different codon in the different genes, but there is no side-by-side comparison. It's the same gene, but specific region has been codon, it changed genome-widely. But as we mentioned, synthetic is we recoded and implemented many PCR tags which change the codon sequence but doesn't change the protein sequences. So this is the best platform actually to investigate the relationship between the codon usage and the translation. Yes, and then we compared. So I'm going to explain how we compared. The PCR tag is around 28 nucleotides. So we can monitor how translation rate change between wild type and synthetic genes. And then we use the downstream region as a negative control. And we can measure the translation rate by the relative ribosome occupancy. We just calculated by the ribosome footprints on the region and then just divided by the messenger RNA. And then by comparing this, what we found is this data. We first see the correlation between the BY4 wild type and synthetic chromosome. And then you can see that each dot is representing the translation rate on the PCR tag. And then you can see comparing wild type and wild type, and then our value is close to the 8.6. But when compared to the BY4 and synthetic, and then this value is decreased always. Only when comparing to the same the chromosome, and then the value becomes higher. On the other hand, in the downstream, meaning it's not changed, it's the same sequence. So our value is higher. So this is the indicating the recorded codon is actually affected the translation elongation. Yes, but the question is, this is really, so any correlation between the codon adaptation index? Codon adaptation index meaning that in higher usage codon is higher, the higher value. So higher codon adaptation index is suggesting a translate faster. So what we're expecting actually that when we change to the higher codon adaptation index, translation also become higher. But actually, we couldn't see any such trend. It's almost like a random. It's showing that actually the change in codon adaptation index doesn't affect it's not a dominant factor for translation rate. And then we use this we demonstrated that synthetic 15 can be a very good learning platform for the fundamental biological questions. And we also did another experiment. Maybe I think people already forgot what we changed is in synthetic chromosome. One of the changes in the synthetic chromosome is we deleted the tRNA from the chromosome 15. So actually the tRNA copy number is drastically reduced in our synthetic chromosome. So I thought we hypothesized that this synthetic yeast has maybe the translation rate is reduced corresponding to tRNA copy number decreased. So we also did this study. So for example, I measure the translation rate on all codons, on all genes, non-chromosome 15 genes, and then compared one by one, and then group by the codon sequences, and then compare the relative ribosome occupancies. So in this case, this is the bar plot is showing the translation rate on all AA codons on Y type and the SYN15. And then can I see any differences? Because actually this tRNA copy number didn't change. And I did this analysis to all codons. And then this is the scatter plot is showing the translation speed between the Y type and the SYN15. Of course, there's no reduction, there's no translation rate change. And then this is the codons, the corresponding tRNA is reduced around 20%, no change. And now 40% also no change, and then more than 40% also changed. So actually, the yeast translation system is very robust against the decrease of tRNA copy numbers. And those kind of thing is very difficult to conduct without synthetic chromosome because reducing tRNA is not so easy, and no one is doing such kind of the rate reduction of tRNA. So we could show that the tRNA synthetic chromosome is very good learning platform for the translation research. Translation research means the protein translation research. Okay, then this is summary. But I want to let me generalize what we learned from the synthetic 15 project. So we want to say that we established the DBTS cycle at the chromosome level. So synthetic biology is always rely on this DBTL cycle, design, build, test, learning, because in many cases, what we design doesn't work as we expected. So the looping this DBTL cycle is always important. Of course, not at the gene level, but chromosome scale level. But we develop Creamier that easily combine and then change the chromosome sequence easily. And this is the accelerate this DBTL cycles. Yeah. So this is the summary of what we are doing. And then now we are also doing something that can accelerate and the change in design and the learning also using synthetic chromosome. Because now we are believing as I showed just now, the synthetic chromosome can be the good learning platform. This is because of compared to gene editing, which can edit several regions of the gene synthesis can change and design change many locus at the genome scale. So I believe that there are many biology that cannot be elucidated just changing few genes. We have to change a lot and at the totally different places. And then there is a biology that can be elucidated only by these approaches. So we are now focusing on those kind of biology now. And then, yeah. Yes, that is the summary of the project and then what I want to do also next. Yes, and then, yeah, this is the team member, actually the old photo, and this is me, and then this she and he and she is the main person of this project. And these are our grantors and the sponsors. Sorry, there is no NIH. Okay, thank you very much for the listening for my talk.
Fantastic talk. This is a huge, achievement, I would say. And also this is important potentially for some other application as well. I want to check with Tuba whether she has comment or questions first.
No, thank you for the opportunity. Great talk. Thank you, Shohei. I really enjoyed it. And
Thank
I
you
don't
very
have
much.
any comments. It's really exciting work. We do fund similar studies at the NIH as well. So we hope to see you as a grantee in the future. And please, feel free to reach out in the future to discuss your ideas.
Okay, thank you.
Thank
So
you
let
very much.
me, ask some questions. So the first question is, from the, NIH perspective. I believe, people in this community done similar work. I mean, first, I believe the E. coli, reduced genome, project, finish, and then we have the now reduced genome E. coli. And as I mentioned earlier, JCBI also did the first synthetic cell creation. And then now we have also minimal, microplasma synthetic cell. And then, a lot of people, international collaboration actually lead to the synthetic, yeast chromosome creation. And then that is actually a huge effort. And then to me, the next thing should be, mammalian cell, synthetic mammalian, chromosome synthesize, and then, make, synthetic, mammalian chromosome, thing. But I haven't seen anything like that because in terms of the, from the NIH perspective, a lot of, biologics, including, some therapeutics, made by Chosel, etc. So that means that it's a very important next step to pursue. But I just wondering why, nobody started, or if I mean, somebody already started, that's great. But I just wondering what is the additional challenges compared to the yeast. Of course, the mammalian cell is much more complex, I believe, but what challenge you still expect from mammalian system, and then how can we overcome that challenge to start mammalian synthetic chromosome project?
Actually, the UK has started to doing the synthetic human genome project recently. See, the Wellcome Trust is, I think, granted some huge grant to UK scientists, I think.
That's what I heard, but that happened this year, I believe, right?
Yeah.
So you
So
are saying now it's time to really jump into the real mammalian system, I guess.
Is there anyone doing it in the US also? The US last time has the human genome right project.
I heard about it, but I never seen any
It's
papers so far.
yeah. There was kickoff meetings and then but
Tuba, do you
it
have any
stopped.
comment regarding, you have some information about that?
Yeah, so what I've heard from the community when it comes to mammalian synthetic biology, the two main challenges are getting things into the cells. The mammalian cells is pretty inefficient, it's very challenging, and especially with SynBio tools, you have large genetic constructs, usually trying to put in whole circuits in there is very challenging. And then the mammalian cells have a way of shutting off things when you put it in there externally as well. So epigenetic silencing is a big issue. Even with like transgenes and like small constructs, let alone whole chromosomes, will be even more challenging. So those are the two big challenges that I've heard from the community. I'm talking to investigators, encouraging them of thinking of ways to optimize their circuits so they can, shrink the size of the vectors that they're using, or, improve efficiency and robustness of the circuits that they're using. Again, this is at the circuit level. I'm not talking about even the whole chromosome level, but there are issues already with mammalian synbio that need to be figured out before we could probably attempt to modify, or make synthetic chromosomes in cells.
That completely makes sense because yeast is very well known for DNA assembly internally. That kind of, facilitate, the entire, synthesis part, a lot. Actually, a lot of people also using yeast for the assembly, including JCBI, chromosome synthesis as well. So mammalian cell is not, in terms of that aspect, it's not, easy to manipulate the huge genome. And the second point is very valid because we still don't understand which gene is actually expressed because the context matters and epigenetics also important. So yeast is probably simpler in terms of that kind of expression.
Yes.
Now I understand, better. So I have multiple more questions, but I see now the 11 a.m. Central Time, so I will close and then I will ask the question later. So thank you all for joining and staying today. We'll meet again on September 18, Thursday, the same time, the same Zoom link. We'll have Dr. Vivek Mutalik at Lawrence Berkeley National Laboratory and Dr. Dimitri Ivancic at Harvard Medical School. As usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in joining us. I'll promote you to panelists who can speak and show your handsome and pretty faces if you wish. And thank you. And I'll stop recording. Just 1 second.